Sweet. Thanks, Stacy. So hi, everybody. My name's Adam Smith. I am on the board of directors for the Rocky Mountain Goat Alliance. I have been a part of the Goat Alliance at some capacity since, right around the COVID year, late 2020, early 2021 as regional rep. Then I decided to get more involved. I was elected to the board last year. I currently serve on the board as a secretary. I'm super excited to be involved with the organization. I'm really excited with kind of the direction we're headed, and I think we've got a pretty high ceiling, and I'm looking forward to where we're going. A little bit about me. I live in Southeast Alaska, little town just north of Juneau called Skagway. I'm an avid mountain goat hunter. I also volunteer as a state of Alaska hunter safety instructor, as well as bow hunter instructor as well. I'm also serve on local fish and game advisory boards, very involved. If mountain goats are at stake, I try to have a way to voice my concern or advocate for them. So that's pretty much how I found my way into the Goat Alliance. I don't really have much else to say. If you're listening, thanks for taking the time out of your night to listen to everything Daria has planned to share with you guys. It's going to be a pretty cool webinar. I'm super excited. And if you're not listening tonight, you're not tuned in, but you're listening tomorrow or in a month or whenever from now, if you're not a member of the Goat Alliance and you love goats, or you're intrigued by goats, we'd love to have you join our organization, and feel free to spread the word. That being said, I'm gonna turn it over to Daria here. Daria, a little bit about her and what you guys are going to hear tonight. Daria Martichenko. She is Dr. Daria. Excuse me. I don't want to rip you of your title there. She is currently working on her as a postdoctoral fellow at Trent University in Canada, studying caribou genomics. And she joined the Rocky Mountain Goat Alliance Science and Conservation Committee in March of 2023. The doctoral research that she's going to talk about tonight focuses on ecology and evolution of mountain goats. And I don't know that I can do much better speaking about genomics and all of those goodies. So I'm going to turn it over to her. Thanks for tuning in. And it's all yours, Daria.
Thanks for that introduction, Adam. Sorry, just bear with me as I try to share my screen.
It's good on my end.
Is it full screen on your end?
It's not. We do not have a presenter mode.
There we go. All right, so today I will be talking about using genomic data to explore the evolution and ecology of the North American mountain goat. So the field of genetics offers a wealth of novel insights to the field of conservation biology. So we can use genetics to discover population structure, estimate population sizes, discover the reproductive strategies, look at population dynamics. We can ID species for forensic applications, and we can ID individuals as well, for example, if we're closely monitoring a herd. So, and we can also look at contemporary and historical population demography. So why are we interested in reconstructing historical demography? So throughout the last 2.5 million years to the present, the Earth has experienced multiple glacial cycles. So climate change has been and is currently one of the main forces shaping ecosystems around the world. And those glacial cycles have resulted in a range expansion and contraction for many species. And we can trace that demographic and evolutionary history in the genetic code of the contemporary individuals. And this is important for mountain goats, as we can see on this map. During the last glaciation, most of the present-day range of the mountain goat was covered by ice. And this is also true is also the case for many species in North America. So by modeling the past, we can understand species biology and predict future responses to environmental change. So getting back to the basics, evolutionary change can be shown by four main processes. So we can have genetic mutation where we have spontaneous mutations arising in the DNA, and those individuals pass it on to the future generations. We can have gene flow where some individuals migrate into population or from a population, bringing or taking away some of the genetic variation. We can have genetic drift. So this is process of random change occurring and frequencies, allele frequencies changing in the population. And then we can have adaptive change, which is the natural selection. So for example, if some of our individuals are better at avoiding predators, they get to survive, reproduce, and pass on their genes to the next generations. So talking about genetic drift, the concept of genetic drift and population size. So this is genetic drift is the random changing of alleles due to random events. So for example, if we have a small population here, we have an unfortunate event that comes and takes out 4 of those individuals. You can see that the proportions of the alleles change drastically. So we're going from 4 out of 10 individuals to 4 out of 7. And if we have a really large population, taking out a small number of individuals does not affect our relative frequencies of each allele. So the effective population size is this statistical theoretical value that quantifies the effect of genetic drift in a single value. So genetic diversity has been recently becoming more implemented into management and conservation strategies for many populations across the North America and worldwide as well. So this effective population size has made it as a genetic essential biodiversity variable. So we're trying to monitor for many populations and species across the world. So the basic definition of effective population size is the size of an ideal population that loses genetic variation at the same rate as the population of interest. So it provides a frame of reference to compare different populations and species. So all populations of species are experiencing different factors that affect offspring survival, reproductive success of different individuals, and this variable can provide us with a frame of reference so we can compare across populations, for example. And the thing to note here, it's usually much lower than the census population size. So census is just counting all the individuals in population. So for example, for humans, we have 8 billion humans on Earth, and NE is only around 15,000. So timescales of NE, we can estimate NE on many from contemporary to thousands of generations ago. So those kind of multiple software packages and analytical methods have been developed to use genetic and genomic data to get at the NE estimates. Okay, so now going to our natural selection, so adaptive change. So the genetic drift and NE was concerned with a random change in population. So for adaptive change, what can we do? How can we assess that in our population? So mountain goat is a unique alpine ungulate. So we have this range that spreads from north to south as well as east to west, and we have multiple variables, environmental variables that change across the range. So for example, here I've plotted annual precipitation, temperature, elevation. So we have some populations of goats that are at sea level. We have some that are really much higher up. And then we have a range of temperatures and precipitations, snow, rain that those populations experience. So there's quite a range of environmental variability across the range. And we also have discontinuous populations. So this is the current day range, and you can see the populations are not continuous, and the dispersal is limited due to the biology of the goats, right? They don't like to cross over lowlands and connect to other populations. So how can we use genomic data? To begin with, I just want to briefly discuss the 2 main types of genomic data that we can use that is used in studies today. So we have whole genome resequencing. So here we just have our strand of DNA, we randomly fragment it and throw it in a sequencer and kind of get our information. And there's also a class of approaches that's called the reduced representation approaches. And the main difference here, we use digestion enzymes that cut the genome at random locations. So that way we're getting kind of a more sparse representation of the genome, so we can include more individuals in our sequencing effort. So we can kind of get a more bang for our buck by having more individuals, but they're more sparsely sequenced across the coverage. And then we can use this sequencing data to see what variants we have. So how is the genetic code of every individual different from another? And to do that, usually requires a genome. So a reference that we can map our reads to, we can map our genetic data to. It's kind of having like a picture in the puzzle box that kind of gives you an idea of where each of the sequence fragments goes on the genome assembly. So this assembly is usually it's the basis for all subsequent analysis. It provides a more robust genomic variation analysis and reduces the genotyping error. So if we know what we're supposed to have in that location of the genome, we have a higher likelihood of calling the variant correctly. So we can talk about discovering loci under selection. We can improve our estimates of inbreeding, annotate genes, so figure out which parts of the genome which function do they do. And then we can also model our historic effective population size. So going back to our NE. And this is then using coalescent techniques. So this is the first part of my research. This is the first paper that I did as part of my PhD. And this is the first genome assembly for the mountain goat. So we released this in 2020. Of note here, and we also made it on the cover on the G3, which is a really cool picture by Kevin White. So the total length of the assembly was 2.5 megabase pairs. So humans will have 3,000 megabase pairs in length. So fairly continuous. And we also annotated the genes, and we've discovered gene families that are unique to the mountain goat. So this provided us with a basis for subsequent analysis. So now we have this reference point. We can look at data from other individuals and figure out where it differs and where the variants are. So today I will be focusing mostly on the results of the paper that we just released last year, that was published last year in Nature Heredity. So this is, and it's titled Contrasting Whole Genome and Reduced Representation Sequencing Approaches for Population, Demographic, and Adaptive Inference, an alpine mammal case study. So I'll be focusing mostly on the mountain goat case study and versus the methodology for the purposes of this talk. So here we've sequenced 3 main data sets. So here they are on the map. So this is the range of the mountain goats. So we have the 5 samples that were sequenced at really high coverage, well, fairly high coverage, 30x. We have 35 samples at mid coverage, and then 254 individuals were sequenced at the reduced representation approach. And from that, we've calculated genomic summary statistics and looked at population structure using principal component analysis. So this this is a multivariate statistical technique that's applied in many areas. It comes from statistics, and it's using multiple variables to condense them into a few that summarize all of the variation. And then looking at the genotype environment association. So we're trying to explain or finding variables that would explain the genetic variation in our mountain goats. So the variables that we used was elevation, then geographical coordinates, and then the climate variation was described by this World CLIM dataset. If you're familiar with kind of studies that use biological, so you can use this for species distribution. This is a fairly widely used data set that combines 20 years of data, and it has 19 biological variables that describe precipitation, temperature, various degrees of temperatures, and so on. So the method itself, so we have our sequenced goats, we have the variation in the goats, so where do they vary? And we're trying to explain that variation with our environmental variables. So this is done using an approach called redundancy analysis. And the good part about this approach, we can have multiple response variables. So we have multiple individuals with multiple variants on the genome, and we'll have multiple explanatory variables. So we can have, with this analysis, we can derive the linear relationship between the two sets of variables. Then talking about population demography. So this is using the coalescent, and I can go into more details about the actual methods. And then to estimate the contemporary NE, so contemporary effective population size, we use this equation by Whipple. So this is using the linkage disequilibrium information from the genome. So to briefly touch on the linkage, so when we have our father's DNA, mother's DNA, and they reproduce, we have those long segments like kind of preserved in their entirety. So here, linkage, so the variants that are in this section are linked together and they're inherited together. So for example, in this child's descendants, we have this little stretch of the pink variants, again, inherited together just because they're closely located on the genome. So, and the further down we go through our pedigree, the more shuffled the pieces become and the less linkage disequilibrium we have as we go through generations. And to summarize, the main point to note here, we had quite a few variants discovered. So we have in the millions for the mid-coverage data set. And to note here is so this pi is the nucleotide diversity. So it describes the genetic diversity of individuals, and it's quite low for all the data sets. So the genetic diversity across the range for the mountain goat is low. Looking at population structure, so every dot here is an individual, and we can see, and this is our RADseq data set, and on the right side is our whole genome resequencing data set. And with the squares, I've noted the genomes that were kind of sequenced at higher coverage. So the AK is Alaska, YT is Yukon, BC is British Columbia, and ID is Idaho. We can see that the genetic variation kind of follows the shape of the geographic distribution. So we have the red samples are the south and the blue is the north, and then same for whole genome, south here and the north kind of in this V shape. And to quantify this relationship further, we can regress. So just using a simple linear regression, we can regress the first PC1 component against the latitude and longitude, and we have a high correlation. So genetic variation, genetic structure is really correlated with geographic distance and location. And for our genotype environment associations, so this model kind of combining all of those climate variables into one group here, and then geography is just our latitude and longitude. And to note here, we can explain 21% of the variation for the RADseq data set using climate and geography, and then 36% for the whole genome resequencing. So quite a bit of variation is explained, but for example, for the whole genome sequencing, we can see that out of the 36% that we've explained, 18% of that is just the coordinates, so just the geography. So again, confirming our previous point from the PCA analysis that the geography plays an important role in the distribution of population structure. And then modeling the historical INI. So this is kind of a dense slide, but I'll try to go over it slowly. So on the bottom here, we have the surface temperature, so just the Earth's surface temperature. And then we have the 4 lines here are 4 individuals at the higher coverage. So Alaska, British Columbia, Idaho, and Yukon. And on the x-axis is our time, so going back in time. So we're started 10,000 years ago, 100,000 years ago, and 1 million. So this is a log scale. And on the y-axis, we have the effective population size. And we can see that the trajectories are fairly similar up until 100,000 years ago. And we can see there is a dramatic drop kind of at the last glacial maximum. So around 20,000 years ago, we have this dramatic drop for all of the modeled with all the 4 individuals. And then we can also use an extension of this approach that models pairwise migration rates between each pairs of individuals. So here we have again, each pairwise comparison corresponds to every individual on the map. And to note here is kind of okay, so maybe I'll revert back. So on the x-axis, again, we have the time going from the present into the past, and the dashed line represents the time point where 50% of the ancestry is shared. So we can see, for example, the furthest back in time is the ancestry that was shared between Idaho and Yukon. So those are the most distantly related individuals on the map. And then the BC and Idaho are closely related, and then Alaska and BC as well. So, and it's also to note here is this approach is one. So the migration rate is almost 100%. So this indicates a fairly ongoing gene flow or a very recent separation. So the individuals are still kind of the same. And then for contemporary NE, we have the quite low numbers. So for our north subpopulation that we've split out, we have from 11 to 10 with 2 different data sets. And for the contemporary south, we have 28 to 24. So this is indicating quite low numbers of NE. So kind of building our low genetic diversity with the summary statistics, the low NE kind of builds and confirms that point as well. Okay, then to summarize, we have low genomic diversity across the range. We have a system that's been heavily impacted by glacial cycles and genetic drift. So we have seen that dramatic drop at the last glacial maximum. That's most likely with individuals being in one southern refugia and kind of spreading out more north. We can see the strong effect of geography on genetic structure. Very low contemporary NE, so low effective population size. And this indicates that it's unclear whether there's enough standard genetic variation for the goats to cope with rapid future climate change. And as an alpine ungulate, they're experiencing climate change at kind of higher rate than, for example, individuals at lower elevation. So this could potentially be a cause of concern for the mountain goats. And with that, I would like to thank you for listening, and then thank all the sample contributors. This work wouldn't be possible without samples that we've obtained, and the hunters have provided the samples as well. And then those are all the funding agencies that provided computational and monetary support for the project. Thank you for listening.
Awesome. Thank you so much, Daria. I don't see any questions in the chat. I don't know if any of our listeners here have any questions. I'm not seeing any raised hands, but now's your chance to unmute. Kevin, I see that you're unmuted.
Yeah. Yeah, I have a question. Yeah, great job, Daria. It's like super exciting to see all the work that you've done. So I guess I had a couple questions. The first one is going back to that slide that you had where you showed when mountain goats split off from the most recent common ancestor, which was like 7 million years ago, I think. It was kind of towards the beginning.
Oh, this one.
Yeah, that one. Yeah, so yeah, I was curious, like, is the idea that like mountain goats would have split off from the most recent common ancestor like somewhere in Asia and then subsequently like migrated to North America across the Bering Land Bridge? Or was that something that happened in North America? Or do you have like a sense of that?
Yeah, I guess we wouldn't get the precise location. Also, this phylogenetic tree, this was based on the UC, so ultra conserved regions of the genome. So it's not like now we can do like full nuclear phylogenies, which we didn't do in this paper. But we can see here that the ranges overlap, the confidence intervals for the split time. So we weren't even able to resolve, because that's a contentious point in mountain goat phylogeny, right? Is it goat the most closely related, or is it the sheep the most closely related species? So we weren't able to definitely resolve this on the tree. And I guess so it's kind of around 8 million plus or minus 500,000 years ago that they had the most common ancestor with the goat and sheep. But yeah, that wouldn't definitely place us on the landscape, I guess. But
Yeah, I was just curious about that. And it did, I'm sure you're all very well up to speed on all the literature. It seemed like I came across like a similar paper on muskox. It was published after yours, and that also might have included, maybe it included mountain goats, just I don't know if it was from their analysis or if they drew upon what you did. I was curious, like, if you were familiar with that one.
is it Aaron Prewers and Chris Kyle paper? Is
I
it?
can't remember the authors. I just remember like coming across it just in the last year and then like, capturing my interest, but I don't really remember the authors, I guess. I wasn't familiar with their names. But
Yeah, the most likely. I mean, all of this data has been released for a while, so and we've shared as well with the muskox group. So if it's the Trent group, so yeah, that's most likely.
Yeah, like so I guess like the kind of follow-up to that is, would you expect like over time as more genomes get sequenced, then it kind of can sort of flesh out these cyber family trees and more resolution?
Yeah, for sure. So with more, actually a person in my lab, in my current lab is, I think she's just in the process of finishing up a paper on the caribou phylogeny, and that's resolving down to ecotype. So we can use, instead of just the mitochondrial DNA or those UCs, like small sections of the genome to reconstruct the tree, we can use kind of the information from the whole genome and go down to like subpopulations or ecotypes. And yeah, we can definitely resolve this tree with much more precision and kind of accuracy. But that's it takes a lot of computational resources just because there's just so many variants, right? Like we have 3 billion base pairs. You compare it between all the individuals.
Yeah. I have another question, but I saw someone else had one too, so like you could maybe answer theirs and I can answer ask mine later.
All right. I saw that Anne, you had your hand up as well.
Shortly. Yeah, thanks. I have a couple of questions as well, so we can tag team back and forth with Kevin and others, etc. Really phenomenal presentation and work. I very much enjoyed it. I'm a wildlife biologist with the Alberta government, and I'm curious whether you or somebody else that you're aware of has scaled it down in terms of the mountain goat genomics to look at things like the impacts of landscape features. I'm not sure what percent of can't remember your exact sample sizes or what percent would have been within protected areas, but I'm curious whether you're aware of any work that is looking at that, particularly when it comes to resistance with respect to connectivity, etc.
Yeah, I guess Kevin, you can comment on this. That's the most of the work on like local herds has been done with microsatellites in Alaska. So regarding the connectivity, I might be another couple of BC herds that also done that. But yeah, to get down to that kind of low level of resolution for genotype versus environment associations is potentially not as informative as just looking at the like relatedness between individuals, right? Because it's at such a small scale, the variation in environment is much less, right? Even for like going back to the analysis that we've done, this is range wide and we're still explaining like 5% of the variation.
Right. And yet, no, I'm thinking of, it's been a really interesting and informative question with respect to wolverines. So that's where that multi-scale approach has been invaluable. But no, I appreciate that. I'll save my other questions for a little later if there's time. Thank you.
And there is one question in the comments, and then we can go back to you, Kevin. So the question in the comments is it worth trying to transplant breeding age goats to different populations to increase genomic variance? For example, swapping some goats from, oh man, I'm terrible at my state acronyms, between states and vice versa.
Yeah, that's I guess difficult question in terms of the management approaches, like which side of kind of are you more of a hands-on management approach? Because a lot of I think the transplantations, like from my point of view, transplantations have to be done very carefully. And we can see, I mean, some of the goats have been transported down south, but it could have unpredictable effects on the current on the existing populations. And given, for the mountain goats specifically, given the low diversity kind of across the range, it's not like we're having a very high diversity spot. Those would be the kind of the source goats to transplant everywhere. I would veer on the side of caution, I guess. I mean, transplantation in general for all species, but also for mountain goats as well. Like I mean, it's more commonly done, or yes, more acceptable to do in my view, if you have a very small critically endangered population, you have to bring individuals. But I think it's better to leave it as status quo for populations, especially for mountain goats, as we don't have kind of this pressing extension concern, which was most likely to be managed by probably managing humans around that population versus introducing new individuals to those populations.
Excellent. I guess Kevin, we'll go back to you.
Yes. The question that I have is some relationship to this question in some ways, or at least to your answer. And one of the things that seems so interesting and has so much potential about sequencing the genome is to be able to potentially identify like genes that are linked to physical characteristics or traits. And I was wondering if in the course of the work that you did, if you were able to identify any specific like trait linked genes.
So no, we didn't get as far as to getting down to what specific genes are. But again, going back to actually this page that we're on, we're explaining a really low percentage of variation with our climate and geographical variables in the system. So the likelihood of finding those genes, I would say, is quite low, because we want to have kind of those really distinct phenotypes for mountain goats. And I'm less aware of kind of like disease resistance, because that's another point, like especially for ungulates now, but like chronic wasting disease. People are looking for potentially identifying those populations that are more susceptible, identifying those that are more resistant. So that could be a potential area of application for looking for phenotype linked genes. But yeah, we haven't gotten that far, I guess, for the mountain goat.
Yeah, it's just thinking like in terms of like localized adaptation, that topic, like, and maybe kind of going back into like a point that you made about how sometimes there can be unintended consequences of translocations, because you might have populations that have localized adaptations that then could be swamped or altered by bringing in animals from a different population. But maybe that sounds like that's maybe a topic that needs more time for there to be work to better understand, possibly.
Yep.
Anne, you had mentioned that you had further questions.
Yeah, does anyone else have a question?
I'm not seeing any other raised hands, and there's nothing else in the comments. So you can go ahead.
Okay, because actually you answered it with respect to answering Kevin's question there in the previous question. I'll just ask a very general one then. What's next in terms of this project or related work?
So I guess I'm not aware that Aaron's doing more work on the mountain goats at the moment. I know a couple of people are finishing up their master's papers with the microsatellites. But yeah, for me, and caribou world now, working on genomics of caribou, which is, I'm sure, and as a management biologist for Alberta, caribou is a more controversial species, I guess, more stakeholders versus the mountain goat.
Understatement, but yes.
Yes.
What would you recommend then, building off this work?
Yeah, I think what kind of the area that I'm currently interested in and working on with caribou as kind of one of the projects is kind of structural variation. So we're looking at kind of point variants in the genome, but we for some species there have been studies where they have identified like the whole section of a chromosome is flipped, and that kind of drives either a migratory behavior or a complete ecotype differentiation. Like social structure in bees is literally just one section of the chromosome is flipped. So that's kind of an interesting point about kind of the theoretical genomics work. And for management, I'm kind of more closely working with Environment Canada now, and then just thinking and looking at the ways how we can use this genomic and genetic research and make it actionable for actual conservation projects and management. Like, how do we bring this, incorporate the genetic and genomic data more into management? Because I think for some management agencies, it's not kind of the first data to be collected. A lot of people just like the hard observational studies or colored animals. So how do we incorporate genetic and genomic data and make it useful for management and make it meaningful?
Good answer. Thank you.
All righty. Is there any other questions? Kevin, I see you're still unmuted.
Oh, no, I didn't have another question. Just
Okay.
forgot to.
Oh, good. Yeah, I'm not seeing any other raised hands, and I'm not seeing anything else in the comments. So I think with that, we can likely wrap up the evening. Yeah, just yeah, we'll call that the end. Thank you so much, Daria. That was a ton of information, and thank you for all the excellent questions at the end from all of our listeners. This is, I guess, my last shout out if there's any other questions.
Good job, Daria. Thanks a lot.
Thank you. Thank you, everyone, for questions. Yeah, and for the recording, my email is at the end of the presentation. So if there's any more questions or people want to chat about Go Genomics, I'm always available.
Excellent. Well, thank you so much. And with that, I will end the recording.